Hello, I am Professor Sims, and in this video I will discuss cell characteristics, properties of light, microscopy, and staining. This is the third of 10 lessons for my Fundamentals of Microbiology course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. Lesson 3 learning objectives are having to do with how light is refracted, how lenses are used to manipulate light, the parts of the microscopes and the different uses of the parts of the microscope, how to do simple stains, differential stains with ground, endospore, acid-fast, negative stains, flagello stains, and all of those kinds of things are going to be covered more extensively in the lab class as well. The theory of spontaneous generation, cell theory, endosymbiotic theory, and germ theory. So this bit is really kind of harkening back to some of the historical things that we learned about in the intro lesson. It goes into a bit more detail. And then, of course, characteristics of prokaryotic and eukaryotic cells. Light waves can be reflected, they can be absorbed, they can be transmitted, okay? And it depends on the property of the material where the light is going through. Light acts differently if it's going through air versus if it's going through glass, if it's going through oil, if it's going through a solid. Refraction is when the light waves are changing speed and direction. In this figure here, figure 2.4, it shows how it's like light is being bent. So it's going through air, which is one type of media. Then it's going through, this is glass, and you see how the light gets bent. It's changing direction and it's changing speed. So when it comes out the other side, it is in a different location. It's actually been bent. It's moved. And this is kind of a schematic diagram. The light coming in is known as the incidence ray. The light going out is a reflected ray. And then the light that is passing through the media is the refracted ray. So it's changed direction, it's changed speed. This is pertinent when you're talking about microscopes because microscopes use lenses to refract and focus beams of light. And this magnifies and resolves images of things that are very, very small. So figure 2.6 illustrates this quite well. It says the lens is kind of like a collection of prisms, and when light passes through a convex lens like this, it's refracted toward a focal point. So light coming from here, light coming from here ends up being refracted into a central focal point here, which is very useful in microscopes. Concave lenses has the opposite effect. So you have a point coming from, light coming from a focal point, and then it's diffracted, it's reflected away from the focal point. So mostly we're looking at convex lenses here when we're talking about microscopes because you want to focus the light and you want to get the light to move toward a focal point. Light itself is actually an electromagnetic spectrum. So different waves have different frequencies and they have different refractive indices. And the different wavelengths are actually observed as different colors. And what's really kind of cool is that the visible spectrum of light is really, really tiny. There are so many other types of light that we can't see at all. You've got your radiation and gamma rays, X-rays, UV, infrared, all this kind of stuff. We can only see this little snippet here. In order to see things like radiation and UV, you can use fluorescent dyes and phosphorescent materials. You can use these things and utilize them to visualize things with microscopes. This here is a bovine pulmonary artery endothelial cells, and they've been excited by ultraviolet light under a fluorescent microscope. These blue things are the nuclei. Resolution power of microscopes. So in lab you will actually be able to quantify the resolution limits of the microscopes. For a lecture, all you need to know is that the resolution can be increased by shortening wavelengths, by increasing numerical aperture of the lens, or by using stains that enhance contrast. So you can either change the wavelength of the light, you can use different lenses that bend the light differently, or you can actually change the specimen, the thing that you're looking at, by adding different colors to it in order to enhance the contrast. And this is just a schematic of the wavelength. So this here is, if this is light, the wavelength is measured here as the distance between, right? Difference. Amplitude is the height of the wave. The wavelength is the distance between one peak and the next peak. And these are different. They're different for different types of light. So this is a low wavelength. This is high wavelength. This is low wavelength. Microscopy, light microscopy uses light to focus a specimen and produce an image. Simple versus compound microscopes, this has to do with how many lenses are in the scope, right? Compound microscopes have more lenses. And then there are several different types of light microscopes. You have bright field, dark field, phase contrast, differential interference contrast, fluorescence, confocal, two-photon microscopes. We use, in the lab, we use bright field microscopes. In fact, it is a compound bright field light microscope. So we have several lenses. These are objective lenses here that have different levels of magnification, different levels of numerical aperture. And we have ocular lenses here. Of course, this is the stage where you put your specimen, and you have your iris and a condenser. These are adjustment knobs. So we're going to cover parts of the scope and things a lot more extensively in lab. But for the lecture, just be familiar with this figure, the different parts of the scope and what they are used for. Electron microscopy focuses electrons using magnets. And electron microscopes can really focus on things that are much, much smaller than what a light microscope can see, because they focus electrons, right? So they go very, very, very small. This is showing the difference between a transmission electron microscope and a scanning electron microscope. The biggest difference really is that the transmission electron microscope, this is all happening in an airlock, like in a vacuum. So you can really get some good, very detailed focus of light here. It's amazing what they can do. Some of these scopes can see things as small as a single atom. It's amazing. Scanning probe microscopy, this has to do with taking, like if you have a specimen, like maybe some muscle tissue or something like that, you can take very thin slices of it and then put that under a microscope and see the cross-section. So this is showing pure gold surface. So this is showing, with an electron microscope image, the individual atoms in a piece of gold. How cool is that? And then this one here is showing plant fibers, right? Very neat. So section 2.4 of the text discusses different ways that you can prepare specimens for viewing under the microscope. The previous slide, we had a look at looking at cross-section, cutting thin sections of specimen for viewing. But now we're going to talk about different staining techniques. So we will be talking about simple stains, negative stains, gram stains, acid-fast stains, capsule stains, endospore stains, and flagellar stains. And each one of these techniques are things that we will be doing in the lab, but we still, for the lecture, you need to know how these stains work and what they're used for. This is figure 232. This is an example of a simple stain of Bacillus anthracus, the agent that causes anthrax. And this has been stained with what is known as a basic dye, crystal violet. Basic dyes stain the cells themselves, okay? And that's a simple stain. Simple stain uses a basic dye to stain the cells. A negative stain over here uses an acidic dye, and the acidic dye is actually repelled by the cells. So the basic dye is attracted to the cells. Acidic dyes are repelled by the cells. So the background in a negative stain is stained and the cells remain transparent. Okay, simple stain, negative stain. This is the process of performing a gram stain. A gram stain is a differential stain, It uses a primary stain, a mordant, a decolorizer, and a counter stain. And it's used to distinguish gram-positive from gram-negative bacteria. We will discuss in greater detail what a gram-positive and a gram-negative bacteria is and what all of that means. Much more detail later on. But a gram stain, it's really one of the most widely used staining procedures. It's very useful for identification purposes, so you should become very familiar with the gram staining procedure, how it works, why it works. Okay, so let's go through this. This is figure 233. First, the primary stain in a gram stain is crystal violet. Crystal violet is a basic dye. It dyes the cells. In fact, it dyes the cells purple, okay? And it will dye all of the cells purple, whether they're gram-positive or gram-negative, okay? It's basically a simple stain in step 1, right? Simple stain with the primary stain, crystal violet. In the second step, you add iodine to the cells. So it still has crystal violet on it, and then you add iodine. This doesn't change the color, okay? But what it's doing is it's acting as a mordant. A mordant is something that is going to hold in that primary stain if it meets certain requirements. So in a gram stain, the primary stain will be held throughout the procedure if it is gram positive, okay? So for this differential stain, and in fact for most differential stains, you know that that microbe is positive if it holds the primary stain. But in order to do that, it does have to have the mordant, and it has to have the required structural characteristics. So in this case, in order for it to hold the primary stain, it has to have the mordant, and it has to be gram positive. Gram-positive means that it has to do with the structure of the cell wall or the cell envelope. Gram-positive bacteria have a very, very thick layer of peptidoglycan. And iodine holds that crystal violet and the peptidoglycan layer together. Okay, so it's kind of like a little anchor. Why does it need an anchor? What's going on in step 3? Well, in step 3, that's the decolorizing step. So in a gram stain, the decolorizer is ethyl alcohol, ethanol. You wash the cells with ethanol, and if the specimen is gram positive, the mordant holds tight to that crystal violet, and the cells stay purple. They stay dyed with the crystal violet. But if it is negative, then those cells will not hold on to the crystal violet, and they'll become clear, okay? So if you were to view the specimen at this step, the cells would either be purple or they would not have any color. And then the last step is essentially another simple stain. You use the counter stain, and the counter stain for a gram stain procedure is safranin. Safranin is pink or reddish. So if at this step you are dealing with a gram-positive microbe, it's not going to be dyed red. It's going to still hold on to that primary stain, that crystal violet, so those stay purple. But if you are working with a gram-negative specimen, going into this step, there was no color. But now it will take on the color of the safranin, the counter stain, because essentially it's just a simple stain. So I know this is kind of confusing, but it's important to know this. In a gram stain, essentially what is happening is you have a simple stain with a basic dye that dyes the cells. Then you have a mordant step that will hold that dye to a bacteria that is positive for what you're looking for, right? But it won't hold it to what you're not looking for. Makes sense, sort of, I hope. And then in step 3 here is really the differential step, because this is where things start to look different, depending on if you have the structural components needed or not in order to be considered positive. And in step 3 of the gram stain, this is the decolorizing step. It's using ethanol, ethyl alcohol, okay? And then step 4 is another basic stain. So if going into step 4 they already have a stain attached to them, then they won't change. But if they don't, then they'll take on the basic dye just like a regular simple staining technique. So it's step 1 is a simple stain, step 4 is a simple stain. And in between you have a mordant step and the decolorizing step, okay? And then at the end, when you visualize the cells, if they're gram-positive, they should be purple, and if they're gram-negative, they should be pink or red. This is an example of how this is a mixed culture, so it has more than one species. These little round purple guys, these are cocci, and they are gram-positive. And then these yellow guys, these are bacilli, and they are gram-negative. Okay. These again are it looks like a negative stain because the background is dark and the cells are transparent, but these are actually capsule stains. And what you do in a capsule stain is, because the capsule is made up of glycocalyx and it doesn't readily take up the dyes. It doesn't take up basic dye or acidic dye because it doesn't have any kind of a charge. They're neutral. So what you have to do in order to visualize capsules is you dye the background with an acidic dye, just like you would in a negative stain. And then you dye the cells themselves with a basic dye, just like you would do in a simple stain. So a capsule stain is basically combining a simple stain and a negative stain. And what you end up with, it's easier to see over here, is a dark background and then cells that have taken on a color. And then you have halos around the cells that are clear. Those halos are capsules. Okay, so this is the background, this is the capsule, and then inside of the capsule is the cell. Over here, it's harder to see because the capsule is so much bigger than the cell itself, but there is a little tiny cell inside of those capsules. In an endospore stain, okay, so an endospore stain is totally separate differential staining technique. But it basically consists, similarly to the gram stain, it consists of a primary stain, a mordant step, a decolorizing step, and a counter stain. And in this case, we have the endospores end up green because the primary stain, malachite green. If the cells contain endospores and they get enough heat, because the mordant in the endospore stain is heat, okay, then the malachite green will be driven into the endospores and it'll be stuck there once the heat is removed. And then you do the counter stain with a basic dye known as carbofuchsin, which is pink. So ideally, and endospore stains are kind of tough to pull off. They are. You'll see when we do them in lab. But ideally, you should have some vegetative cell left, or what is known as a mother cell that's undergoing sporulation. That will be pink. And then the endospore itself should be green. These are flagellates, bacteria that have flagella. In fact, these are specifically known as paratrichous, which means that they have flagella all around the perimeter of their cells. Flagella stains are kind of difficult. I mean, flagella are so thin, and it's really, really hard to get them to dilate to the point where you can view them under a microscope. So it takes the use of heavy metals and some nasty irritants, but it is possible to get nice flagellate stains. I think we're going to talk some more later about different types of flagellates, different arrangements, and things like that. But for now, we're going to have to back it up a little bit and talk about history. So the theory of spontaneous generation. This is like when we were talking about in the intro class with Hippocrates in ancient Greece. They used to believe that it was possible for life to arise from non-living matter. And I mean, some smart people believe this, including Aristotle. But scientists were torn on this for a long time. John Needham was arguing in support of spontaneous generation. Lazzaro Spallanzani was one of the early contenders where he was arguing against spontaneous generation. Francesco Redi in the 17th century, so back in the 1600s, he was able to show the first significant evidence that refuted spontaneous generation because of his famous experiment where he showed that in order for maggots to develop on rotting meat, okay, ew, gross, But in order for the baby flies to start growing on meat, there had to be access to the meat by adult flies. In other words, they had to lay eggs, right? And then, of course, Pasteur, we talked about him a little bit earlier. He was able to conclusively disprove spontaneous generation with his very famous swan neck flask experiment. And he was the one who was quoted as saying life only comes from life. And of course he was right. So figure 3.2 shows the famous maggot meat experiment. But yeah, essentially it was, if the container was open and flies could get in there and lay eggs, then baby maggots showed up. If the container was sealed, nope, nothing happened. And then in order to, because people were like, well, yeah, because there's no oxygen. Okay, so let's put a mesh and oxygen can get in, but flies can't. Guess where the maggots ended up? Where the flies could land and access, right? So figure 3 shows some of these guys we were just talking about. This is Reddy, Needham, and Spallanzani. Okay, so those were kind of some heavy hitters when you're talking about spontaneous generation. But the big enchilada was Louis Pasteur's swan neck flask experiment, and this is figure 3.3 from your book. Essentially, there was 2 parts to the experiment. In the first part, he had broth in the flask, okay, and it has, broth has things in it that that bacteria and fungi and things they love. So if there's anything in the air or anything at all that could get into it, then they're going to go in there and reproduce and grow and grow and grow, right? Well, we know that now, but this experiment really kind of showed the difference. So what he did is he sterilized the broth. He heated it until it was completely sterile, and then he left it open to open air. Except the design of this flask meant that it was essentially closed off so that the outside contaminants can't get into the broth. They could get as far as here, and essentially they were stuck, right? So he did it that way, and he was able to demonstrate that nothing grew in the broth. There was no spontaneous generation in the broth, and there was no way for outside contaminants to get in there, so it made perfect sense to him. Then he did the experiment again with the same flask, the same sterilization technique, and then he broke off the swan neck so that it was totally open to outside contaminants, and wouldn't you know it, they got into the broth and grew like gangbusters. So it's like, yes, you need to have life in order to create life, right? Throughout the years following that, we had many more developments. In the 17th century, of course, we have talked about Robert Hooke already in the intro lecture. He was the one that was able to first visualize cells under a microscope, and he was the originator of the science of cell biology. And then, for hundreds of years, couple of hundred years, scientists were debating each other, talking about the nature of cellular regeneration. Where do they come from? Are cells the fundamental unit of life? And eventually, cell theory was popularized in the mid-19th century. The tenets of cell theory, as they have been adopted and are used today, are that all living organisms are composed of one or more cells, and that the cell is the basic unit of structure and the basic unit of organization in organisms, and that cells arise from pre-existing cells, right? We've already talked about binary fission, and so figure 3.5, this shows the first observation of cells by Robert Hooke, right, via a microscope in the 1660s. But it took another 200 years for cell theory to be widely accepted. And it took the work of many scientists. These are just a couple. This is Rudolf Virchow and Robert Remak, right? So these were proponents of cell theory in the mid-1800s. Endosymbiotic theory is the theory that says that mitochondria and chloroplasts So these are organelles, right? Subcellular organelles. They have their origins in bacteria, and this is supported by significant structural and genetic data. Of course, the genetic data came much later. So have a look at figure 3.7. I'll just go through it really quickly. It says that, way back, like early Earth times, you had what was known as a proto-eukaryote. So they were just beginning to get to the stage where they were forming nuclei. And then there were infoldings in the plasma membrane of the ancestral cell that gave rise to these endomembrane components. So this is where you started to first see features that are found in modern eukaryotic cells, subcellular structures, organelles, and things. And it started with an infolding of plasma membranes. And then in the second endosymbiotic event, early eukaryotes consumed photosynthetic bacteria. So first came the mitochondria, right? And then came chloroplast. And the idea here is that they actually consumed photosynthetic bacteria, right? So photosynthetic prokaryotes. And then they formed chloroplasts. It's really kind of cool. The germ theory of disease states that many diseases are caused by microorganisms which invade humans, animals, and other hosts. Right. So this is pretty much common knowledge now. Germs are bacterium, protists, fungi, virus, prions, or viroids. But it was first proposed in 1546. That seems like a long time ago, but not really. Girolamo, I'm sorry, I don't know how to pronounce that name. Fracastoro in 1546. And then it was 200 years later expounded upon by Plinsick. And this whole idea was rejected by many scientists for many, many years because they were still holding on to this theory from Galen, miasma theory. It translates roughly into bad air. Scientists and doctors of the day believed that people became sick because of the air. There was pollution in the air. There was nasty things in the air from like rotting meat and sewage and stuff. And that's how everybody got sick. And, now we can think of airborne pathogens, but they did not believe that there were living things in the air. They believed that the infection came from the air. Okay, so there became a shift in ideas in the 1850s. Right along the same time, they were discovering that life comes from life, and developing cell theory was becoming popular, and this ended up culminating in the golden era of microbiology, which we have talked again. A lot of this is review from the intro lecture, but so eventually the germ theory replaced the miasma theory, right? And again, we have Joseph Lister and Koch and a whole bunch of other prominent scientists to thank for that. Well, now that we know that life comes from life and we know how to visualize cells and all of these fun things, we have learned quite a lot about not only the structure and function of modern cells, but also their evolutionary history. So let's talk some about that. We'll go into prokaryotes first. Prokaryotes are the more ancient types of microorganisms. They don't have subcellular structures. They don't have the organelles, membrane-bound organelles. They do have things, they do have a cytoplasm. They have ribosomes, that they can have DNA plasmids, and they usually have a nucleoid where the genetic material is held. They have a plasma membrane or a cell wall. Some of them even have capsules. They can have flagellae, but they do not have membrane-bound organelles. They do not have membrane-bound nucleus. Some prokaryotes have cell walls, and cell walls are really useful in not only in maintaining the cell's structure, but also protecting them from changes in osmotic pressure. And I will have you look through the isotonic, hypertonic, hypotonic. That should be some review from maybe chemistry. Just have a look through that and familiarize yourself if you have forgotten. equilibrium and things like that. Endospores, this is not something common to every prokaryote, but some prokaryotes can form endospores. Endospores are survival mechanisms, so they're kind of like hibernation for the cell. If the cells are in environments where something has changed, the food is gone, or there's been a shift in temperature or the pH, or there is some kind of inhospitable environment, the cells can sporulate. They can turn from regular living, dividing, reproducing cells known as vegetative cells into endospores. And I would have you familiarize yourself with the steps, but essentially it starts out similar to binary fission or budding, where the DNA replicates. But then you have like a separate membrane that forms around the DNA. Then you have a four spore that forms, and you have like a layering of additional membranes around this replicated DNA. And then essentially that layer, it builds and builds and grows until you have a cortex that forms, and then a protein coat is added to that, and then an endospore is released. So it has this really big, thick protective coat around it. And inside of that spore is genetic material, and it is there, and it stays there, and it's viable, and it's just kind of waiting until the conditions improve. And when the conditions do improve again, that spore will germinate into a vegetative cell that will then reproduce and grow and thrive again. Prokaryotic cell envelopes, they include a plasma membrane. The plasma membrane is composed of phospholipids, and it is a phospholipid bilayer with polar heads and hydrophobic tails. They have integral and or peripheral proteins. A lot of prokaryotes also have a cell wall. The carbohydrate components of the glycoproteins and glycolipids, they actually extend out from the surface of the cell, and all of these components can vary greatly between different species. I would have you look through the different types of transport across the bacterial membrane, and I also will direct you to some videos I have in the playlist that can help illustrate this as well, beyond just reading it in the textbook. Now, when you're talking about prokaryotes and their cell membranes and things, so here we come back to gram-positive and gram-negative, and it's important to understand that gram-positive and gram-negative prokaryotic cells are named for basically this difference in their structure. Okay, so gram-positive cells, they have the regular plasma membrane, right? And then this is inside of the cell, the cytoplasm. And then they have this cell wall that is made up of a very thick layer of peptidoglycan and teichoic acid. Okay, gram-negative bacteria. This is the inside of the cell. This is the plasma membrane, right? So far we're looking the same as gram-positive. But then here is the peptidoglycan layer for a gram-negative cell. See how it's so much thinner, right? And then what is this? There's another plasma membrane. So this is the fundamental difference. Gram-positive bacteria have one plasma membrane and a very thick layer of peptidoglycan. Gram-negative bacteria have two plasma membranes that sandwich a little thin layer of peptidoglycan. And they have these lipopolysaccharides, lipidase, a lot of other things going on that gram-positive cells do not have. Some prokaryotic cells produce things called glycocalyx capsules. So we've had a look at capsule staining already. So this is a capsule stain where the background is dark and the cells are dark and the capsules are surrounding the cells. These capsules protect the cells. The ones, some prokaryotes make them, not all, but they protect the cells from things like desiccation, right, from drying out. They aid in attachment, so you see capsules a lot in your pneumonias, right? That's what makes them sticky. It makes them stick to your lungs. And then your flagellates, these are the different types of flagellates. So you have the monotrichous or monotrichous. This is where they have one flagella at one pole only. Amphitrichous means that they have one flagella, but they have one at each pole. So one at this pole, one at this pole. Lophotrichous means that they have more than one flagella, and they can have more than one flagella at either one pole or both poles. And then paratrichous is where they have flagella all around the perimeter of their cell. Some prokaryotic cells also have fibria, which are used in attachment, and pili, which are used to transfer genetic material from one cell to another. I would like for you to go over these concepts in your book in greater detail, okay? And flagellates, we'll be looking at flagellates in, I think it's lab 5. Eukaryotic cells have membrane-bound nuclei and they have membrane-bound organelles. They generally have 2 lipid bilayers and that regulate transport of materials into and out of the cells, which we have talked about different forms of transport already in this presentation. The nucleolus is responsible for ribosomal synthesis. The endomembrane system includes the membrane-bound organelles, vesicles, endoplasmic reticulum, and Golgi apparatus. The smooth endoplasmic reticulum it's for lipid biosynthesis, carbohydrate metabolism, so breaking down starches and carbohydrates, and detoxification. The Golgi apparatus, those are the ones that process proteins and lipids. They produce glycoproteins, glycolipids, and those are important because remember, glycoproteins and glycolipids are components of plasma membranes. So if you're going to make new cells, you need to make new membranes. Golgi apparatus play a big role in that process. Lysosomes, that's where the digestive enzymes are made. So they break down ingested particles via endocytosis, and they break down larger particles using phagocytosis. And lysosomes also break down any kind of intracellular components that have been damaged. The cytoskeleton is made of microfilaments and microtubules that provide structural support and can also serve as a network of transport, especially if you have microtubules that have made up flagella or cilia, right? Centrosomes are central to mitosis, okay? And we're going to talk some more about mitosis and cell division in a later lesson, but centrosomes are things that form the spindle in mitosis when cells begin to divide. Mitochondria, you guys know mitochondria is the powerhouse of the cell. So they do the cellular respiration, the production of ATP, energy. And then, of course, plasma membranes contain, in eukaryotic cells, contain sterols, glycoproteins, good Lord, mumble mouth, glycoproteins and glycolipids. And here's a figure. This is from your book 335. So if you were to look at this cell and you look at a prokaryotic cell, obviously eukaryotic cells, they're much larger. They're much more complex. They have much more moving parts. Okay, so it's a very big difference. Figure 339 is showing the nucleus and the nucleolus. Okay, so in the eukaryotic cells, these are 2 different things. The nucleolus is inside of the nucleus, and this is an amazing electron micrograph that is, I mean, you guys, that's tiny. It's so small and it's so clear. It's like a miracle of science. Anyway, geeking out a little bit, sorry. The centrosomes, right? They consist of centrioles and microtubules, and they make up the spindle that's used for mitosis. And then, of course, you have your mitochondria. I mean, look at this. Electron microscope,
image of a real mitochondria. That is amazing. It's just amazing. And then the flagella and cilia, just like everything else in eukaryotes, flagella are going to be much more complicated than they are in prokaryotes. They have a specific array of microtubules. They move differently, right? They can rotate and roll, and they can whip back and forth. And then paramecium, you may have seen some of the paramecium in our pond water experiment in lab 2. These guys they're just an example of eukaryotes that actually have cilia all over their body. So yeah, they don't just have, flagella at the poles or something like that. They actually are covered in cilia, which is why they are so fast. They swim so fast.
So that was lesson number 3. I want to thank you guys for watching. Don't forget to do the reading. Check the description below for more videos related to these topics, and leave your questions for me in the comments section.